Welcome to Zoom O'Clock with your host, Tessy Antony de Nassau. This podcast brings you enlightening discussions with leading experts and public figures directly to your ears.
Hello, everyone, and welcome to Zoom O'Clock with your host, Tessy Antony de Nassau. I have a very interesting guest for you today. I'm talking about Dr. Ruslan Rust, comes originally from Belarus and grew up in Germany. He moved to Switzerland for his PhD at the ETH in Neuroscience. Now he is a group leader at the University of Zurich and works in the field of cell therapy for brain regeneration. I have as well another dear friend next to me, Claude Camarena. I always pronounce it wrongly. And she's joining us as well, for all the way from Beverly Hills. And let's see where we're going. I have prepared some questions for you to start off the conversation. Are you ready, Ruslan?
Thank you, Tessy, for the kind introduction. It's a pleasure to be here. It's my first ever podcast, so I'm excited.
Wonderful. I'm so happy to have you. And, at the dinner last time at our house, I was just fascinated with everything you were saying. And I said to my husband, I need to get Ruslan on the podcast. For everyone listening in and looking at our video on YouTube, Ruslan and I met because of his girlfriend, Katie, who has been the midwife delivering my baby boy, Theodor, in the Hirslanden Clinic in Zurich. So a very medical affair and really good friends now. And I'm really looking forward to what you have to tell us, dear Ruslan. So my first question to you, it might be a bit controversial, as you have put it as well, when we were talking about the questions, because still in the 21st century, it is being used and it is needed in some instances, and yet there's a very, very big lobbying against it. A lot of people are fighting against it. And I'm sure that some people listening to this will not be happy to hear about it, but it is important to learn about why we do the things we do in order to understand them. So Dr. Ruslan, your view on animal experimentation and use of animals on neuroscience, please.
Yes, as you mentioned, it's a very important and controversial topic. And I find it also sometimes a bit sad that because it's so controversial, most people discuss it very emotionally, right? There's often, it's not really a neutral conversation, but it's, yeah. Maybe I can shed some light on how it works from a scientist perspective, because I believe many people just know it from the outside, or they just saw a picture or no effect. So in generally, in Switzerland, before you start any experiment that might require animals, usually apply for a license. And the license, how it works, it usually weighs the potential benefit to the humans against the distress that is caused to the animal. So this is the general principle. I believe this is quite important because what many don't know is that probably a large share, or 95% of the experiments we do in the lab, they do not involve animals. Mm-hmm. So the general rule is if something can be shown in a computer model, or if something, can be shown in a petri dish, something that you want to find out the efficacy of a drug or so, they also do it like that. And even if you use animals, then there is also the rule that you use the lowest animal in the hierarchy that you can use to show the effect that you want to see. And I think this is something very important, because many people think that if you do research, you use only animals for all the experiments, and that this is simply not true. It's just if you have a special drug candidate that you found out of many others, you tested it in cell cultures, you modeled it, there comes a step where animal models are required. Yes, and I believe, maybe for clarification, that many people also slightly they kind of think, or they expect too much from AI or a petri dish. I mean, the technology we have there is really far, far away to model the physiology of the human body or of an organism in general. I mean, sometimes I say that, we probably will be on the Mars 50 years before there will be a replacement for animal models. This is really how far we are away to really understand all the physiological components and what is going on in the body.
Very, very interesting. No, and it is really, really important to say that as well, because as you said, there is an emotional aspect of it when people talk about it, and it is important to understand the science behind it. And as you said, 99 point, I don't know how many percent are not, are tests which are not using animals. And when we're talking about animals, you talk about little mice as well, I assume. And so yes, very, very interesting, especially as well the component on the AI, as you said, we are not there yet. And also the vaccines, such as the COVID vaccine that we have needed to be tested on a living organism at some point before being injected in human beings. Exactly.
I believe there is, so I'm not a really expert in that field, but I would say that there's probably almost no drug that you can, that went through the FDA approval and so on, that was not tested on animals. Yeah, it's one of the steps that you do some quality controls in the phase before you start the clinical trials that require model organisms. And maybe if I can add, this is something very important, what you said, that we use mice. So generally, it extremely depends on the field you're working on. I know that some people, for example, that work in bone regeneration, a mouse might be a really bad model because the way their bones are assembled is completely different than to us humans. Maybe a sheep could be a better model, or I'm not an expert in there. But whereas for us as neuroscientists, I believe the biggest part of the experiments is done in mice and rats, so in rodent animals, because their brain has some striking similarities to the human brain.
Very, very interesting. Wow, I didn't know that. When I worked in the P3 lab on influenza and HIV/AIDS, when I was doing my thesis on biological terrorism in Luxembourg at the Luxembourg Institute of Health, I was doing that internship. There, I saw as well they were doing tests with guppy fish, because guppy fish are the only fish that can regenerate their lungs and pieces of heart by itself. When you cut a piece out, it grows back by itself. And so they want to see which genes, which strands of DNA that makes that happen, so that they can maybe at some point replicate that for human beings as well. Kind of like you see in the Marvel movies where a hand is cut off and it just grows back, right? How fascinating. So let's move over to the next question, because you don't just talk about mice, you do so many other things as well. So we put our models with human brain cells becomes every year more complex, right? One day, as you said with AI before, maybe how about consciousness, right? So the ethical challenges in neuroscience, what do you have to say about that?
Yes, this is, I believe, this topic gets more and more important, and sometimes it also gets neglected, and it actually also affects my research, because we work with mice, as also most or many of the neuroscientists, but our cell therapies are with human cells. Yeah, so these cells have a human origin, and there are experiments in biology that are clearly controversial, because, for example, one very famous one is if you take, for example, a pig embryo, you can add some human cells to it, and then you inject it, or you transplanted it to a pseudo-pregnant pig, and what you will get is an embryo that is mostly a pig, but it will have, like a mosaic, some human cells. Yeah. And obviously it's not allowed to that, they give birth. So there is a certain stage in the pregnancy where they need to kind of euthanize the animals. But that this is controversial, I believe everyone can understand, right? And the main reason why it is controversial is, I believe, the same reason why our research or neuroscience in general becomes also, let's say, ethically, there are some open questions. This is because what happens if the human cells, they go into the brain? Yeah, it means that at some point, people might think that if you build a complex neural system, it might become something like conscious and might have like maybe, if you would go really far, it could have some type of human rights, or I don't know. This becomes for us more and more a topic because all these models, you've probably heard of that, the organoid models, the cell culture experiments, they start having more and more, let's say, aspects of a human brain. Mm-hmm. And maybe what was recently controversial is, so they recorded, I believe, some brain activities in these organoids that resemble these of a premature baby, which was quite a big claim, but of course this raises a lot of attention, media attention, and the ethical debate started then.
Wow, that's incredible, because that goes as well. So in your research as well, I know it is not related to neuroscience, but one thing that was really big in the media, right, with using animals was what was the name? Daisy the sheep, or what was she again?
Dolly the sheep.
Dolly, exactly. Mm-hmm. Wow, with the pigs, I have never heard about that though, and I can understand that there must be some people.
I can add something to Dolly if you were interested in that, and many people don't know that, and I was very surprised. But so this was, I believe, the first cloning experiment. So they cloned Dolly from a different sheep, and I mean, this is done regularly right now. If I'm not 100% sure, but I think this has been done with cats, with dogs, with monkeys. So the whole cloning procedure is actually, I believe, it's almost commercially. So you can clone your cat. I believe, I mean, not in Europe, but there are probably countries where you can do that. So this field of research also continued.
That's crazy. It's a bit like there was these science fiction movies. I don't know if you have seen them, where when your cat or dog dies, you take the cells and you put it into the cloning, and they clone your animal that just died, kind of thing. So incredible. So you think that they are? Yeah, well, it makes sense. Behind closed doors, everything is still continuing, and people just don't talk about it. But
I mean, yes, I believe. These aspects, I think there should be a very clear ethical kind of border, and that should be discussed by the experts. And I mean, how it is in Switzerland, you can't really do these experiments behind, or in some shady place, because all this lab equipment you require and all the funding, I mean, usually this needs to be public, or it needs to be at least publicly announced. And I have the feeling that maybe there are countries where there are some secret projects ongoing. I don't know about that, but usually there should be ethical borders, and then the scientists should, I believe, kind of adhere to that. And then we have an agreement what we can do and what we can't.
Absolutely. So also with what you can and what you can't. When we had dinner here, I don't know what we had as a food, but when we had dinner, we were talking about, so we have a mouse here at home. Her name is Cinderella. She's a white mouse, and we saved her. Julia actually said, Tessy, I want to have her, because we saved her from a snake in an animal shop in Luxembourg, from being dinner to the snake, I mean. And so he said, oh, that's similar to the ones we have in the lab. That's how the conversation started. But what was really interesting, what you were saying, is that, the animals do not suffer, which is really important, I think, as well, to say, there's specific ways what you're doing, and And can you explain us a little bit about that, the procedure? What really, just because it's interesting, and I'm sure that people are actually thinking about that, what actually happens in the lab to a mouse like that?
I think this is also a very important point, because when we sometimes have also days where people can visit animal facilities, or you can have a visitor who comes with you. And the reaction I always get is, wow, that's so clean. And that's so, I thought this would be completely different. And I need to say that in Switzerland, we have very high standards. And even within Switzerland, there are facilities that are more modern, where probably the animal welfare is even better than others that might be a little bit more, traditional, let's say. And this is very, very important for us, because there's also research interest, not only the empathic way, but there's also research interest for us that the animals are not suffering, because we want to have very controlled conditions. Ideally for us is that if every animal is exactly the same. Because when we start testing a drug, we don't want that there is an effect because, for example, an animal had an inflammation or some other, or it's stressed. Stress can also cause, we nowadays, all types of effect on the body, on our physiology. So we are also extremely interested in that they are well, that they eat enough, they drink enough, and are not stressed. Generally, for everyone who's interested, I believe there are ways to enter like these facilities and to see for themselves how it works.
Very, very interesting. Well, as I mentioned to you again at dinner last time, I will definitely come and check it out, just because I'm fascinated about the topic. And I am an animal lover at heart. I really am. As a child, I had 10 million animals: hamsters, mouse, snakes. I had worms, I had dogs, cats, cows, chickens, like you name it, I had it. So I really do love animals, but I do as well appreciate the science for the medications that we are using all as human beings as well, and that animals are being used for it. Until we don't need to, right? As you said before, we don't use, you don't use animals because you want to, you use the animals because you need to, in order to keep us safe, right? In order to make sure that we have the medications that we need as human beings in order to be healthy, right? So until we have an alternative, this is what it is, right?
Yes, and I maybe add one last point, because I think it really fits nicely to the conversation. So we have in Switzerland, and I believe it's actually not only in Switzerland, it's probably a European organization, which is called 3R Competence Center. And this 3R Competence Center, so 3R stands for Reduce, Replace, Refine. They actually provide scientists the funding to improve the animal welfare. So, for example, and this should be done through innovation. So we are actually funded also by this organization, because I can give you a practical example. When we transplant the cells in the mouse, then we might be interested in the survival of our cells. So we want to see how are the cells doing in the mouse, and want to look maybe at different time points. We want to see after 1 week, after 2 weeks, and after 3 weeks, how many cells are still there. Did these cells, change to or differentiated to other cells, and so on. Now, if you would do that, you would need to euthanize to every time point a certain amount of mice that you require for this time point. And what we, for example, develop is some type of an in vivo imaging method, where our cells start glowing, and this glowing we can see through a camera. It's based on it's like the firefly, the glowing of the firefly. And because our cells are glowing in the mouse brain, we can look at the same mouse to different time points, which means we save a lot of animals, and we can longitudinally suddenly look at our cells and understand how many of them are still there, and how many might have not survived. So I believe these organizations, they also show that there's another way of improving animal welfare. It's not only by saying you don't perform any experiments anymore, but it can also be through bringing in new methods that improve the whole situation.
Mm-hmm. Wow, really interesting. I will definitely look up reduce, replace, refine and put it as well below, because I think that's really, really important. So my next question. So you work in cell therapy, as you mentioned, for brain injuries. Why do you believe that cell therapies have enormous potential and are superior to normal drugs, especially in the brain? But also, why do you still have no cell therapy?
Yes, of course, that's a good question. Yeah, why is something so good that it doesn't exist? Yes, so I mean, of course, it's a huge topic that one could write the book probably about, but I just tried to keep it simple and concise. So generally, a stroke is a heterogeneous disease. Yeah, we have in a stroke, we have this reduction of blood flow. We have inflammation, we have scarring, cell death, and so on and so forth. So all these mechanisms are very different. And if you just add a drug that, for example, a drug that is a neuroprotectant, that protects neurons from dying, you still have maybe 4 or 5 major pathological mechanisms that are still ongoing. And most likely this one drug, targeted against one of let's say, the problems, will only give you limited success. And to be honest, this is also what we see. So there were many drugs for stroke in the field of neuroprotectants, or they interfered with the inflammation, that were not successful in a clinical trial. Now, the special thing about the cell therapy is that generally a cell can work on with different mechanisms. So there's this one, I would say, relatively intuitive mechanism, is that if there is a damage in the brain and you transplant the cells into these damaged areas, they might kind of form new neural circuits and repair some damaged function. However, this is not the only mechanism, but we also know that if we transplant cells, these cells actually can also provide many, or they release many factors. We call them cytokines. And these cytokines also provide a big help for the endogenous tissue to repair itself. And this is almost the more interesting mechanism, or it might potentially be, because there you, of course, can have pro-angiogenic or pro-inflammatory factors, which suddenly do not affect one certain mechanism, but kind of affect the whole area in itself. And maybe if I can add why there are no cell therapies available.
The question is burning.
Yeah, if it's such a magical tool. So there are a couple of major obstacles in cell therapies. So first, I actually not sure what to start with, but I think the most intuitive one is, which cell should we use? Yeah, I mean, we have an injury in the brain, so we want to have some type of a neural cell, most likely from the nervous system, that might replace that. Now, these cells are not easy to generate. Where should we get them from? In the past, it was done with primary tissue. So you looked at, or you took from abortions, you could take some fetal cells. And if you were assigned to a clinical trial, that could be done. But there's a limitation of these cell sources, as you can imagine. Mm-hmm. And this, for example, this limitation we are overcoming right now, because we can now get neural cells also from adult tissue. So you can generate it from your own, from, for example, your blood cells. You can do it with a process that's called reprogramming. You can kind of bring every cell of your, or not every, but many cells of your body into stem cell-like phase, like an embryonic stem cell. And these cells you can then differentiate into neurons and other cells. So this would be solved. Yeah. But the other question is also, how do you transport, or how do you bring the cell to the region you're interested in? And I believe this is currently quite a big problem, because of course you can transplant the cell directly through the brain, which is relatively invasive. Mm-hmm. And many
dangerous
patients
many patients, they don't make it, no?
Yes. So, I mean, the procedure itself most likely extremely depend on where the stroke occurred. It's probably easier to transplant into the cortex at the surface of the brain than to go into subcortical structures that are more in the deeper areas of the brain. But this is, of course, I mean, a stroke patient, many of them have functional deficits, but they're not terminally sick, right? It's not that they have nothing to lose. So that's why ideally you would have a systemic injection of the cells through your vein or through an artery. And this brings you then the issue, how should the cell know where to go, right? It would circulate in your whole body, and how do you make sure it doesn't end up where you don't want to have it? Mm-hmm. And maybe a last limitation, you always, with these cells that you generate, the question is, where do they come from? If it's a cell that is generated from you, this is immunologically, I think, relatively easy. So there will be no problems of an immune incompatibility. But it's logistically super difficult. Yeah, so the ideal cell therapy would be like an off-the-shelf therapy, so you have some type of universal cell line, you take it out, and then you can transplant it to any patient. And currently, we still have problems to make like a universal cell line. So the problem is, if I would donate a cell to you, these cells, like with organ transplants, similar to that, would be rejected. Yeah, so you might want to try immune suppression or something like that, which is again not ideal. So on all these questions, we in our group work to improve that. Mm-hmm. And to kind of bring the cell therapy more towards the clinics.
Very interesting. I have, for me, with cell generation in the brain, well, I've never experienced a stroke, right? But I had a huge depression and burnout 6.5 years ago. And something I did was RTMS therapy, which is also known as repetitive transcranial magnetic stimulation, to kind of like rebuild the neurological pathways which have been lost due to anxiety and depression, which is kind of like a non-invasive therapy, which I'm sure you're very well aware of. So I wanted to mention that just for the people listening to it, if you have something such as a burnout for the brain, that has something that has worked for me very well. I don't know what you think about RTMS therapy. And then just going through what you mentioned before with you cannot really transplant the brain cells from one person to the other, like we would, for example, with the microbiome in the gut, right? Because there's already these scientific advances, and I have invested in a company in Bavaria who is doing that, where you kind of like use the microbiome of a human being to kind of find out cancer early. And at the same time, through the microbiome, you can implement the microbiome of one person to the other, and it automatically changes that person's gut. So if you are anxious, if you are depressed, anxious, or overweight or whatever, with the transplantation of that microbiome, you give the person inherits that as well, that you translated it or transplanted, if you want. So that is really fascinating when I heard about that, because of course that is the negative side effects. If you get to transplant of a person who is not healthy, that is not the way you should do it. So of course they make sure that it's a healthy person. But isn't that incredible, right? Because there it works. So how long do you think this is going to take for the cell transplantation of the brain to actually work?
Maybe I can add also quickly to your microbiome comparison. I think the major difference there is you don't really, so in the microbiome, you can correct me, maybe I'm wrong, but I think there usually you have the bacteria and like this is a mix of non-human cells, right? Whereas when we transplant cells, this is of course human cells, and your immune system reacts differently to it. In the like for the microbiome in the environment, there are bacteria, so the whole immune system is adapted to that. Whereas if you bring in like unknown cells that are recognized as foreign cells, then your immune system is activated.
With the brain, and sorry for interrupting you, but with brain, is there not some immune suppressants that you could take? Because you have them as well for other things, for cancer therapy, for example, you have these immune suppressions, or when you do like organ transplantations of a liver or something, you get specific medication that the body doesn't eject the strange organ from someone else. Do we have that for the brain?
Yes, so actually there's currently a cell therapy trial in Japan, if I'm not wrong, where they do it for Parkinson's. So they transplant Parkinson cells that were generated similar to how I described we generate our cells. And they actually do exactly this, what you said. Yeah, they give a strong immunosuppressant and bring the cells to the brain. Now, what adds? So this is a bit a philosophical question, but in my view, I want that the human stays the same and I change my cells. Yeah, so I try to make my cells, they are kind of perfectly adapted to the human I want to transplant the cells to. And I'm also more working with genetic and molecular tools. So it's more, let's say, my expertise or my passion that I find interesting. I find it not convenient that the patient needs to take immunosuppressants. I would prefer to have a safe cell that is not recognized by our body as foreign. But on the other hand, I mean, this I also need to add, it's also risk, right? Because if your body is not able to recognize a cell as a foreign cell, and then something strange happens to the cell, so it turns into a cancer cell, which can theoretically happen, your body will not be able to recognize it, right? So this is also a safety aspect. But there we actually also work on, like, it's a genetic safety checkpoint, so we can use a drug, and when we inject that, then the cells are immediately gone. So to your question, so it's possible to use immunosuppressants. In my view, it's not very convenient, but from a clinical perspective, yeah, it's done.
Okay. Wow. Okay. We have we have passed our time many times now.
Oh, I'm sorry.
No. I love
Too long
it.
answers.
I still have questions, so I think we need to do another podcast, which we will talk offline for sure. One thing I ask all of my guests is there a reading that you would suggest for people to go and get if they want to learn more about this? Or a podcast that you're listening to, other than, of course, Zoom O'Clock, that you would suggest to people if they want to learn more about these topics?
There is a very interesting book. It's about the history of, but let me quickly check the exact name, the history of the brain, or we maybe add it to the show notes. Let me see. I found it. It's called The Idea of the Brain. And it's a very fascinating book because it tells you a bit the history, how people start understanding what the brain actually really does. And there was for many, many years, this discussion, is the heart, historically the heart is the most important, but the feelings come from the heart. And they show how slowly which processes had to happen that we attribute now to all these, cognitive feelings and so on to the brain. But it goes kind of from the past to the present to the future, and also what will be maybe one day possible to do. So I extremely enjoyed that. It's by Matthew Hobb. is the offer, and maybe we can just link it on YouTube or in the show notes. We can add.
That's for sure. Thank you.
Yeah.
Thank you. That is amazing. Any podcasts before I let you go? Any podcast you would suggest for the nerds out there like us? Other than Zoom o'clock, of course.
I need to admit that I usually try to listen to podcasts with non-scientific topics. Science is already like 8 to 10 hours a day in my head.
Exactly.
So which if I want to relax,
give us your nerd podcast or something fun or eccentric that you're listening to. What is your type of shutdown of science and get back to some other stuff?
There is, this is probably very mainstream, but these true crime podcasts is something that I don't know why I'm interested in that, but it's always entertaining and to listen to that.
Thank you. Well, really, really nice. A specific name of one, maybe?
Actually, no, several. We can have a look later. I need to check that, which ones.
I put it in the show notes then. And then, so Dr. Ruslan, thank you so much for your time. That was really interesting. I have still so many questions, so there will be a number 2 of this when you have time again. And
anytime.
Please say hello to Katie as well from us. And thank you. Thank you so much.
Yeah, thank you for inviting me. It's also a pleasure. I saw you had so many amazing guests. That's good to, or it feels nice to also join them. And anytime, let me know when we should do a follow-up if people are interested, and I will find time.
Perfect. Also for the people listening in, if you have further questions, please do directly message me on Tessy_from_Luxembourg on my Instagram, or over Twitter, Tessy_DE. And we will collect the questions for you for a next podcast with Dr. Ruslan. Also subscribe, like, comment, rate, and share, because only like that will we reach a wider audience and can inform and entertain people from all over the world about new and exciting topics, such as the one we have discussed today. So thank you so much again. Thank you, Claudia, as well. Claudia, do you have anything else to add? Because we were so into this. Is there anything that is burning?
I felt like I have so many questions but thank you so much. I really wanted to know what you would think in terms of combining the general public in wellness and cell repair. What you could share to the general public that you found interesting, that maybe from a scientific perspective would be very, very beneficial.
I believe in general that what for the public might be very interesting is that the application of cell therapies is extremely diverse. So there are some people who say there was always this first generation of drugs where you used, like paracetamol and so on. So you have like small molecules and they do something very like unspecific. Whereas now there is this time of antibodies or biomolecules. You probably also heard of that. And this was kind of the next generation, and it helped to cure or to at least help a lot of people with many diseases that were untreatable before. And many people say that actually gene and cell therapies will be the next generation of treatments, because you have so much more potential compared to like a single drug. And it can be that it will take way longer than we expect. This was also our experience with cell therapies. There was already a promise 20 years ago, so we need to be patient and hope that there will be a big potential.
That's amazing. Thank you.
Thank you.
Well, thank you, Doctor. So that will be it for now. I know you're very busy, so we let you go now. You're off the hot seat, and we will reconvene once I have collected more questions for you for podcast number 2 with Dr. Ruslan from the University of Zurich. Thank you so much for your time, and have a wonderful rest of the day.
Thank you. You too.
Thank you for listening to this Zoom o'clock. We hope this discussion was insightful and has provoked some new ideas for you. Please share and subscribe. If you like to keep in touch with your host, you can find her on Instagram under Tessy_from_Luxembourg and on Twitter under Tessy_DE.